So bacteriophage elements, these bacteriophage elements, they are what? They are mu elements, which is an unusual phage which integrates into the host genome by the method of transposition, not by site specific recombination. Okay, it has the genes for transposition also, for the packaging also. Next is eukaryotic transposon. Around 10% of the eukaryotes have retrotransposons, transposons, and generally most common one is retrotransposon. They are like IS element only. Next is P element, which is the transposable elements in Drosophila, having 2907 base pair long element and 31 base pair inverted repeat at each end. And they can encode transposase enzyme, transposable element, and they do not utilize RNA intermediate during transposition. They can insert themselves at many different positions in the Drosophila chromosome. And this P elements, it's a causative agent of hybrid dysgenesis. What is hybrid dysgenesis? It refers to the high rate of mutation in the germline cells of the Drosophila, which results from a cross between males having P element and the females which lack P element. So this syndrome is marked by temperature dependent sterility, elevated mutation rate and increased chromosome rearrangement and recombination. The mating between females from N strain. Now if you mate them, the mating between the females from M strain of Drosophila with males from the P strain results into sterile F1 progeny. Okay, P strain into M strain, it results into sterile F1 progeny. P strain is which have P element. Okay, the mating between females from M strain. Okay, yeah, the reason is that P strain contains numerous copies of P element which are mobilized in the embryos derived from M eggs. Okay, so it enters into M eggs. The P elements enter into M eggs, the embryo of M eggs. So these eggs lack a repressor protein that inhibits P element mobilization. Okay, as a result of which P element transposon reside passively in P strains because they express a repressor that keeps the transposon silent. So in P strain, why this is not expressed? Because P strain have the repressor as well. It have the P element plus the repressor. That is why they do not express in male. But when they are mated with an M strain lacking such a repressor, the transposons are mobilized and it often integrate into the genes required for germ cell formation. And this explains the high frequency of sterility in the offspring from such crosses. Very interesting, right? So when if you reverse this, when M strain is male, that means male do not have the P element but female have the P element. In that case, F1 progeny is normal. So try to understand that. Hybrid dysgenesis manifest when crossing P strain males with M strain females and do not manifest when crossing P strain females. Why? P strain females are those females having P strain with M strain males. So the eggs of P strain females contain high amount of repressor protein. So wherever P element is there only repressor protein will also be there. So when eggs have P strain, they would have repressor also, which prevents transcription of transposase gene. So the eggs of M strain mothers, which do not contain the repressor protein, they allow for transposition of P elements from the sperm of the father. So if the father have the P elements, the repressor M strain do not have the repressor. Okay, the M strain are the ones which do not have the repressor. So if repressor is not there in the female and the father P element is released, it will be mobilized into the embryo, as a result of which the progeny will be sterile. But in the reverse case, when M strain is male and P strain is female, in that case female also have the repressor protein. In that case, what will happen? It will be female P strain will not be expressed, female P element will not be expressed, as a result of which it is repressed and there will be normal fertile progeny. So this was about P element. Next is ACTS element.